Thank you all for joining us for the February 2024 monthly seminar on physical genomics. We're very excited to have as our guest today, Dr. Erez Lieberman Aiden of Baylor College of Medicine. Before we get to Dr. Aiden's talk, just a quick word for those of you who have not joined us before. We're coming to you from the Center for Physical Genomics and Engineering at Northwestern University. We're a transdisciplinary research center focused on studying the structure and function of the genome through the convergent science of super resolution imaging, modeling, computational genomics, biology, and artificial intelligence. We have a dual mission to develop new strategies for the treatment of disease and the reversible engineering of living systems, and to train the next generation of multidisciplinary scientists and bioengineers. You can learn more about our center and our research and our training programs at physicalgenomics.northwestern.edu, or you can follow us on our YouTube channel where we have an archive of our public seminars and lectures, and we're also on Twitter, X, and Instagram. As I said, we're very excited to welcome Dr. Erez Lieberman Aiden today. Dr. Aiden is an applied mathematician and a molecular biologist. He's associate professor at the Baylor College of Medicine. Among his many achievements, of which I'm sure many of you are familiar, he conceived of the Hi-C protocol, which is today in widespread use in most genomics projects. And he assembled the team that created the first 3D map of the human genome. He's won numerous awards, including recognition for one of the top 20 biotech breakthroughs that will change medicine. He's received the President's Early Career Award in Science and Engineering, the highest government award for young scientists. And his laboratory has been recognized on the floor of the U.S. House of Representatives for its discoveries about the structure of DNA. Dr. Aiden, it's an honor to have you share your work with us today. For our audience, you can ask questions during the Q&A at the end of the talk, or just type them into the Zoom chat, and we can ask Dr. Aiden at the end. So with that, Dr. Aiden, I can turn the floor over to you, and we can get going. Thank you so much.
Thank you so much. I'm really honored to be presenting here. I hope, can people see my screen okay?
Yes, looks great.
All right, terrific. Yeah, it's a real honor to be presenting. I was just commenting about how exciting it is, that there are such things as seminars on physical genomics, which is not something that would have existed even a decade ago. So it's really an exciting time in the field of genomics, and especially in its intersection with physics. Today, I'm going to give you a kind of quick intro to sort of some of our work in this space, but I really want to focus on some of our recent work on fossils of ancient chromosomes, because I think it's quite remarkable that this works at all. There's a lot of things in science where you kind of figure it should be possible to do it, and then there's sometimes where you really have no clue at all if it's possible to do it. And well, this is one of those stories, the latter type. Can people see the sort of top, the titles and stuff? Is the share screen stuff including that in some way?
No, I think it looks fine.
It looks fine. It looks fine for you. Okay, terrific. Okay, so there's a lot that we know about the shape of DNA. If you look at 10 letters of DNA, it looks like a double helix. We've known that for about 70 years now. And so that's good, but there's actually a lot, there's a lot to learn, despite the fact that the discovery of the double helix really led to the birth of molecular biology. There's a lot that we don't know. And just to give you a sense of scale, I mean, relative to the scale of the whole genome, which can have, or a whole chromosome, which can have hundreds of millions of base pairs or even billions of base pairs in some organisms, 10 base pairs, the scale of double helix, that's roughly kind of like the scale of, the silicon atom to the size of your cell phone. Right. So if you say, well, my cell phone is just made of silicon, now I understand its structure, you would clearly be missing many intermediate levels of the game. And I think that is true for the genome as well. There's a lot that we don't know about the shape of DNA. How do the 2-meter long genomes, when you stretch them out from end to end, how do they pack into a nucleus that's typically a few microns wide? Really, really interesting basic question. I'm sure you guys are very familiar with. And your body has many cells with different functions. And if you look at them, the genes that are on in a cell, they're actually controlled by the shape of the cell's DNA. So for instance, this has been recognized since the late 70s that there are elements in mammalian genomes that live very far from target genes, but seem to be able to turn them on or off. People were skeptical about this for the first few years, but then realized it was happening, named these things enhancers, and hypothesized that they functioned via three-dimensional loops. That even though they're far in 1D, they're actually close in 3D. And so what this highlights is that the 3D packing of the genome is not purely a problem for physicists, right? It's a problem that's incredibly relevant, intimately related to the function of the cell, in the biological sense. And so there's a problem that has a considerable amount of urgency, I would say, for multiple fields. We can study the shape of DNA. We're very good at studying the shape of DNA, as I'll show you. I mean, kind of compare it to our ability to study other shapes. We're very good at studying the shape of DNA at this point. And one of the methods for doing that is a method called contact mapping, which is a way of exploring structure via 3D proximity events. So, and look, I want to be truthful, right? The best way to explore the structure of things is just to look at them. If they're far, you look with a telescope. If they're small, you look with a microscope. And, this has been a very, very powerful engine for the forward progress of science. I mean, one can argue that in some sense, the telescope and microscope really catalyzed the scientific age in which we live. I do not know what the future brings. But, in 500 years, if aliens arrive on this planet and say, take me to your leader, I don't know. Maybe we will say something to them like, oh, leader, what is that? We live in an anarcho-syndicalist commune. But if they say, take me to your microscope, I'm fairly confident that we will be able to say, oh, here it is. And here's the latest improvements. Because this is an incredibly powerful technology. Contact mapping is a bit, it's a little weird, right? It's this other way that you can look at structure that seems right now to have an advantage over the microscope. So I'm going to tell you a little bit about it. You may know, and I apologize if you are already fully aware of this, but I want to make sure everyone's on the same page. I'm going to be showing a lot of pictures that are going to look kind of similar, so I want to make sure people understand them. So let's think about contact mapping in the abstract. Here's the most recognizable person in the world, as best I can tell, based on trying to give versions of the slide in different places. Homer Simpson, he lives in the fictional town of Springfield. And let's suppose that we wanted to understand the 3D structure of the Springfieldians. We could look for events of extreme proximity. Here he is in extreme proximity with a donut. Here he is in extreme proximity with another individual. That's interesting. More data on extreme proximity, more data, more data. Finally, I have a slide full. At this point, I need a data management strategy. You can imagine different data management strategies, but the one that I'm going to use for the data on this slide is to put in a matrix. This matrix shows the frequency with which two individuals are in the same image in the previous slide. And as soon as you look at this, you realize, well, I might not. Can people see my pointer, by the way? Can people see my pointer?
Yes, we got it.
Okay, great. So you can see that, this individual and that individual, they seem to be up to something. They're in contact very frequently. And indeed, they've nucleated a whole family of individuals with an enhanced contact frequency. And that is quite curious and quite interesting, and highlights the sort of vexing nature of this data, which is that you can identify patterns. You don't necessarily know what those patterns mean. Anyway, that would be a total waste of your time, other than the fact that we can do this for the human genome. We have two little bits of the human genome that are in close proximity. We can read that out using a sequencing assay. This was developed by myself and Nick Van Berkom and others as my PhD thesis back in the late 2000s. It's, based on, many, many decades of work, really going back to the development of DNA ligase and restriction enzymes, where some of these types of experiments were used to probe the flexibility and bendability of DNA, already going back to the 60s by cutting the DNA and ligating it. And then people started gradually were able to do this better and better inside living cells. in the 80s and 90s, and finally with the development of the human genome, it became possible to do this kind of genome-wide. Especially once you had, the human genome and high throughput sequencing, it became possible to develop these sorts of technologies where you could do this genome-wide and look at contacts coming from all over the human genome at once. And Hi-C really, I think, was first technology that really enabled looking at these kinds of contacts genome-wide, but building on a very long stack. Anyway, so I'm not going to go through all the full details here, but roughly speaking, the way that this works is you sort of stick the whole genome in place by cross-linking. You cut it into little bits. You fill in the ends of the bits with biotin to facilitate extracting them later, and then you ligate the bits. And what's happened when you ligate these bits using T4 DNA ligase typically is you take two sequences that used to be close in 3D and you made them close in 1D. And now that they're close in 1D, you can read them off with a genome sequencer, which is great because we're pretty good at genome sequencing these days. And so then what you do is you pull down these ligated bits and you sequence them. Typically, it had been done in the past using paradigm sequencing, and that's still very common today. Anyway, so that's it. It's kind of cut and paste, cut, paste, down. This generates genome-wide contact maps. So the human genome, for instance, comprises 23 pairs of chromosomes. Every bit of any of those pairs of chromosomes can be, in principle, in contact with every other bit. And in practice, at some frequency, they are in contact. And so you get a contact map that looks roughly like this one. This is just sort of a typical example. There are 23 bright squares along the diagonal, and those correspond to, as you may guess, the 23 chromosomes. And what they are is manifestations of a physical property of the chromosomes, namely that human chromosomes, and this is typical for mammalian chromosomes and chromosomes of many other species, they aren't fully stretched out in the nucleus. They don't look like when you're cooking your spaghetti, each spaghetto is all over the pot. No, they're not like that. They sort of fluctuate within distinct subvolumes, the nucleus, called chromosome territories. And for that reason, loci that are at opposite ends of the same chromosome have an enhanced contact frequency as compared to random loci, which manifests as these 23 bright squares along the diagonal. I'll note that these matrices at the highest resolutions that we interrogate them, if I printed them out at, a millimeter by millimeter scale, they would, occupy like all of Asia, which would be illegal. So it's hard to visualize them. And so one thing that we've developed these, interactive visualization systems for being able to zoom in and out of these matrices. They work basically like Google Earth, and we developed is the first one of these in 2014, and now there's other similar systems that a number of groups have developed. But anyway, if you, for instance, scan this QR code, it will take you to this particular map in an interactive form where you can explore it yourself. And I'll have a few of these QR codes in the talk. And so if you see a QR code, you can scan it and immediately use the real data yourself that you're browsing in and modifying and exploring to agree enthusiastically or object strenuously to my bizarre interpretation. Anyway, the power of this is a very big matrix, right? So we can zoom in. So here I'm going to zoom in on chromosome 8, and you can see that there's this very, very sort of rich kind of pattern on the chromosome. The most striking feature is this bright diagonal. Now, why is that there? It's there because loci that are adjacent in 1D, because they're tethered together by the polymer chain, they can't get very far in 3D, and so they tend to form contacts with one another. And so the contact frequency of two loci skyrockets as the loci get closer and closer on the same chromosome, which leads to this bright diagonal on this chromosome and every other chromosome in every other successful Hi-C experiment that you will ever see, or at least that have been seen so far. But that's not the only feature, right? You can see this interesting sort of checkerboard pattern, right? Plaid type pattern where you sort of see one pattern, another pattern. What's going on with that? This is actually the pattern of, one pattern is actually associated with active loci, open chromatin. The other is associated with inactive loci, inactive chromatin, closed chromatin. And they seem to occupy two distinct subvolumes of the nucleus called the A and B compartment, because when we came up with this, we gave it the most boring possible name, and people subsequently have come to me and say, well, why the hell did you name it something so darn boring? And I feel a little bad about that. And it was a failure of nomenclature. Hopefully we'll do better later in the talk. But anyway, that's the A and B compartments, the segregation of open and closed chromatin in the genome. And what you're highlighting is that even at the very, very top level, right, the ways in which the activity of the genome shapes its 3D architecture. And just make a note of it because we're going to come back to it later. So in these plots, as it is always the case, pure white means no contacts. Pure red is some sort of maximum here at 700. So the intensity of the map is showing the contact frequency between any pair of loci in the genome typically at a resolution. Here we've binned the genome into 250 kb bin. So this is 250 kb squared for each bin for this particular map. Now, we didn't see any loops in the first maps, and this was an embarrassing situation for us because the only reason people actually cared about the 3D, like there weren't seminars on physical genomics at the time that I suggested this. As I was working in Eric Lander's lab, and I said, Eric, we should work on this problem. It's interesting. Let's try to develop an assay for probing how the genome folds via sequencing. But the thing is, there weren't things like seminars on physical genomics. So no one was the least bit interested in that problem. Like that was like not an obviously interesting problem. I mean, although it was an obviously interesting problem, but like what's obvious is so sociological. So why would anyone be interested in doing this? And if that was the selling point, right? The selling point is you could figure out genetic regulation by finding the loops. And that's always been on some level the selling point in this business to biologists is really hard to figure out cell regulation. If you could physically see the regulators and the regulatees, catch them red-handed in 3D, gosh, that would be great. And so the big problem with the early maps is we didn't see any loops. Why would you expect that we could see loops in such an assay? Well, if you imagine two looping loci, they should form lots and lots of contacts. And as you move away from them in 1D, the contact frequency should decline. And so looping loci should be really, really obvious in a high-C contact map. But we didn't see any loops in the first maps because the resolution was terrible. This is a map, looks pretty good maybe, but it's a whole chromosome. The resolution is 1 million DNA letters. So if you printed them out with a pixel, each pixel being a square millimeter, it would cover a large conference room table. Which is actually how I used to visualize these things before we had juice box. I would just print them out on a large conference room table, and it's fun, because you're like doing this weird thing, and if you're like leaning into that as your social persona, then like being the guy who's like looming over all this printed out stuff on a big conference room table, like it kind of works with that. So anyway, I would do that, and it was a good visualization system. But our resolution was 1 million DNA letters, and that's just not the scale at which genetic regulation works. We couldn't see a darn thing. So we developed a better method. So that original method published in 2009 called dilution high C. Now Suhas Rao and Marion Huntley, two students in my lab, said, well, that stinks. We gotta improve the resolution. So they came up with something called in situ high C. We can do this whole thing, but instead of doing it in massive dilution, which had been suggested by, for instance, Joop Dekker's chromosome conformation capture experiments and protocols, they said, hey, maybe, you don't want to do this in massive dilution. You don't want to blow the chromatin to bits. Maybe the loops are very delicate. Let's try to prevent the loops from breaking up. We'll handle them very gingerly. We won't lyse the cell nuclei. We'll figure out how to do everything in nucleo. And so developed in situ high C. And that coupled with improvements in sequencing, and this is how it always works. You have to improve your protocol. You have to improve your molecular biology and your biochemistry, and then you also have to, increase the sequencing depth. They go hand in hand to enhance your resolution. And so now I just want to show you like what did that enable. So here's the first high-C map that we published at chromosome resolution. It looks good, but if you zoom in some more, here at the bottom, each pixel represents all contacts between one kilobase of the human genome and another kilobase of the human genome. Here we're now at the scale where a gene is an appreciable size, right? You're looking at a whole chromosome, a gene is trivially small, right? But now we're at the scale where genes are appreciable sizes, genetic regulation events should be happening, but of course the map doesn't look like anything. There have been a number of efforts to improve this, but generically they didn't really change the overall situation, which is that things look good at the chromosome scale, but did not look good at the scales at which genetic regulation happens. But then in situ high C made it possible finally to have maps that looked pretty good at the scales of genetic regulation. For the first time, you could clearly see loops, right? These very, very bright peaks that we hypothesized about. I do want to note that I've kind of covered this in about 20 minutes right now, but this took 7 years. including 5 years after the publication of the first IC paper. And it just highlights, sometimes it just takes time to get things to work, right? I mean, we just like kind of hammered at this, and I, give tremendous credit to Suhas and Miriam for just hammering at this problem for 5 years until they finally were able to get to a point with the experiment where you can see loops. So that was good. Now, the resolution here is a kilobase, 1,000 DNA letters, or about one micron in the of DNA if you stretch it out from end to end. So if you print this out with each pixel at a millimeter squared, the map would cover the Texas Medical Center, or Dallas downtown, or many American cities. Anyway, this resource was shared. It's been used in, I don't know, 6,000, maybe it's like 8,000 other studies now. Anyway, people use this data a lot. And we observed 10,000 loops. Nearly all of them were between convergent CTCF sites. That was very striking. People sort of thought that CTCF was involved in looping, but it was very, very striking what we observed. When we looked at these loops and zoomed in, you could see that the loops seemed to correspond to the positions, for the most part, the correspond to positions of the protein CTCF and RAD21 and SMC3, which were associated with cohesin. Cohesin complex was well known for its role in metaphase, but it was pretty unclear actually what it was doing in interphase, although it was known that it tended to be around like enhancer sites. Anyway, you look at it, you see the CTCF, the cohesin, okay. But then we saw also something that was totally shocking. I actually didn't believe it for a long time, and I kept giving, this computational science in my lab, I kept giving speeches about how, look, you should never show me stuff that's obviously an artifact. Just find the bug in your code, because certain things are just not reasonable. Anyway, I didn't think this was reasonable. So CTCF, so cohesin basically goes where CTCF goes in interphase to first order. That's an oversimplification. Cohesin, will stop at other sites, but let's set that aside for a moment. Mostly goes where CTCF goes, and CTCF doesn't go places willy-nilly. It binds to a particular motif. Now, the human genome is double-stranded. So that means that any DNA motif, any DNA pattern can be written on one strand or the other. You can think of it as it can either point forward or point in reverse. So if we look at a pair of CTCF motifs, such as was associated with every loop, in principle, the motifs can be in 4 configurations: the convergent orientation, both pointing towards one another; the divergent orientation, pointing away from one another; the forward orientation, both pointing towards the Q terminus; or the reverse orientation, both pointing towards the P terminus of the chromosome. And by dumb luck, you would expect each of these to occur about 25% of the time. But in fact, we observed overwhelmingly that all of the loops were between convergent CTCF sites. And that was very, very, very weird. Extremely hard to understand. I mean, I want to give you a sense of scale. If you blow everything up by a factor of 1,000,000, the CTCF protein is about the size of a pea. The loop links a length of DNA, it could be a kilometer long, right? How in the world do these proteins figure out if the motifs are pointing at one another, pointing away from one another, right? It's like inconceivable. Anyway, so we decided we need to come up with theory. Here's what we came up with. We said buried in the nucleus on the DNA fiber, there's this extrusion complex. We hypothesize it exists, and it has 2 subunits, and it lands on DNA at adjacent sites, and then the 2 subunits start sliding in opposite directions. Now, when I'm driving, I don't drive forever. Eventually, I stop. And when do I stop? I stop for stop signs. But I don't stop for all stop signs. You only stop for a stop sign if it's pointing towards you. So now look at this top subunit of this hypothesized extrusion complex. Oh, it's coming up to a stop sign, a CTCF motif, and the CTCF motif is pointing towards it. So it better stop. It's going to stop. Now, the bottom subunit, it hasn't seen a stop sign, so it's not going to stop. Here's a stop sign, wrong direction, still not going to stop. Here's a stop sign in the right direction. So we hypothesize, oh, it's going to stop. Now, if such a machine existed, if the extrusion complex existed, then it would explain the convergent rule. And we thought, hey, cohesin, it's donut shaped, maybe it slides. Although, it wasn't really known at the time that, that cohesin could slide along the genome. Of course, now it's well known. It was unclear, right? This would imply that a large loop starts out being a very, very small loop. That's a little hard to understand, because if you believe in things like the 30 nanometer fiber, which today we don't really, don't think is ubiquitous, but 2014, 2015, people thought that that was ubiquitous. Then the chromatin is much too stiff to form that initial loop, that initial tiny loop. And so there were a lot of big questions about this. And so we did a lot of CRISPR experiments to explore this. I'm not going to be able to get into that. So we did a lot of CRISPR experiments to explore this and explore the predictions of this theory and extensive physical simulations. And finally, we proposed this is happening due to interphase loop extrusion. I should note that Kim Naismith had proposed, for instance, that SMC complexes could be involved in loop extrusion during metaphase. When we read that, we felt a little more comfortable. When we put this out 2015, thought, who knows? That turns out to have been a pretty good prediction. I should note that other groups, like the Murney group subsequently, like Leonid Murney's group, subsequently also published a paper based on the convergent rule, hypothesizing the same thing. So, and beautiful points about this made also by Victor Corses's lab. The convergent rule is very, very suggestive, essentially, that such a mechanism is taking place. And there's been a lot of beautiful subsequent work showing that this is essentially how things work. Now, I want to point out that studying the shape of DNA is not just a way to understand regulation, but it's a way to understand other life forms. And I'm going to take a step back for a moment, okay, to the sort of problem of genomics, which is sequencing a genome. And sequencing a genome is that joke. There's that joke. I know all the digits of pi. Oh yeah, well, prove it. That's impossible. You can't know all the digits of pi. Oh yeah, sure I do. 1, 2, 3, 4, 5, 6, 7, 8, 9, 0. There it is. I got them all, right? Because the problem, it's not knowing all the digits, it's putting them in order, right? And the same is true of the human genome, right? Knowing all the bases, that's easy, right? Putting them in order, that's the hard part. So how's that done? And basically, all genome projects are done more or less in the same way. You take a DNA sample, you make many copies of it. It's a little bit different now. In Oxford Nanopore, sometimes you can do it a bit differently, but broadly, you collect a sample of DNA, you make a lot of copies, you break them into little pieces, you sequence the individual pieces, and then the hard thing is you take out these little pieces, you have to put them in the right order. And this is actually what cost so much time and energy for the Human Genome Project, which spent decades doing this at the cost of $4 billion. And this, even up till the early mid 2010s, still cost millions of dollars to get those sequences into the correct order. But we actually have been working on this problem for a while. And other groups had actually published some early work on this, although it didn't lead to a reliable solution. But really nice work from the Shinturi group and from the Decker group, also kind of looking in this direction. But there wasn't really no solution for this. And I just want to highlight, this is the front page, actual front page of April 1st, 2016 New York Times. This is not the COVID pandemic, but the previous Zika pandemic. And the genome that everybody wanted was the genome of the Aedes aegypti mosquito, which was the principal vector for the pandemic. And it was known that the Aedes aegypti mosquito had 3 chromosomes, but this was the state of its genome. It was all in tiny little pieces. No one knew how to put them in order. And it's because, there was like 15 different ways of doing, trying to do this kind of thing, but none of them worked reliably at all. And it was millions of dollars, years and years of work to do this kind of thing. Anyway, fortunately, Olga Dudchenko in my group had been working on the following sort of concept. So Hi-C makes it really clear when things are in order. You see here, we're looking at a nice Hi-C map where all the chromosomes are in order. It's all great. We feel great about it. But now look at this. This is a Hi-C map like I haven't shown you before. This is for a genome that's not in order. And all of a sudden, there's little blocks all over the place, far from diagonal, et cetera, et cetera, et cetera. And Olga said, look, if I look at this Hi-C map, if I develop the right algorithms, the right kind of machine vision, although we didn't do this by machine learning, but we do some of that, but today, but she just developed, the old school machine learning algorithms for doing this kind of thing. She said, look, if I, look at this thing, I should be able to just algorithmically figure out what order the genome is in. And this took a problem that in principle was still cost millions of dollars and years and made it easier. Let me show you how much easier it made it. So this is attempting to put together the genome of barrel clover. You can see it's a hot mess. This is this is about 2015, I'm going to say we're doing this. Genome's a hot mess. Every technology, long reads, every single thing, optical maps, all of this stuff had been used to try to put this thing together. It was still a fragmentary mess. So Olga generated about a couple $100 worth of high-C data and had developed an interface on top of our Juicebox browser to try to put it together. So let's watch her do that. It's fun to do it on top of Tetris music. So what you can see is she's taking the little segments of the genome and she's just putting them in the right order. And just first, she's just gathering the segments that belong on the same chromosome by looking for those off-diagonal blocks that are the telltale sign that two things are supposed to be close in 1D, but haven't been put close in 1D. So she's gathering all the chromosomes. You can see this in real time. The clock is iterating. This is sped up tenfold. So it's about 11 minutes of her time total. She's still gathering these chromosomes. And then once the chromosomes are gathered, she's now putting each chromosome into the right order and orientation. She's just going chromosome by chromosome, doing this, we're I think 7-8 minutes into this. And, she's got most of the chromosomes done. She's got the last two to do. And here we are after a total of 11 minutes. And you can see that the chromosome has been properly assembled. So that's 11 minutes of work and a couple $100. This is a problem that at the time was costing groups millions of dollars and years of work. Anyway, we had all kinds of plans about how to publish this. It was clear after that story appeared in the Times that, it would be completely unethical for us to do anything other than to immediately release that genome. And that's what we did. We released that genome a few weeks later, a few weeks after it appeared on the front page of the Times, correctly assembled into 3 chromosomes. Just to highlight what happened on this. So, for Aedes aegypti, we didn't do it using that interactive approach, but instead we used Olga's automated 3D DNA pipeline, because there's a lot more pieces. So even if you have really great music in the background, you're not going to want to do that manually. So we took that fragmentary genome, it was on the front page of the Times, and via process of correcting misassemblies, iteratively scaffolding, and merging overlaps, totally automated way, produced this nice 3 chromosome assembly and solved that problem entirely. And this ended up published in Science in 2017. It then did a better version, showing that if using long reads and showing that you could do an even better version in 2018. Together with the latter was together with a large group of collaborators. And actually in 2018, based on all of this, we launched a collaboration called the DNA Zoo Consortium to make 3D genome assembly widely available, because this was just so enabling for genome projects. And today, basically every genome project uses Hi-C for assembly. So if you were working in doing modern biology in essentially any species, you're on some level using Hi-C data, using some of the methods that we and others popularized. And many institutions have actually participated in this DNA Zoo project. So one of the goals is to share the methods, but another goal is to simply share lots of genomes, especially focused on mammals. And so there's many institutions that send us samples. If you send us a mammalian sample, we will assemble and release it at no cost. I think we've so far released 3.5% of all mammalian species. We release them at dnazoo.org without any restrictions. You can use them, do whatever you want with them right away. And, like the Zoonomia project, for instance, which published like an issue of Science last year, they used like 100-something of our mammalian genomes. We're happy for anyone to do whatever they want with them. About 9% of mammals are in our pipeline. If you've got a mammal we don't have, please send it to us. Finally, I want to turn to the final and actually main theme of the talk, which is that studying the shape of DNA is actually, because we're so darn good at the shape of DNA, it's actually a way to understand the past in ways that I didn't fully appreciate until recent years. So we asked the following question. We said, look, what happens to geometry of chromosomes after a long time? What happens after a long time? So what do I mean by a long time? Here's a woolly mammoth. It died, say, 52,000 years ago. I have one in particular in mind. And, over time, it spends a lot of time, sort of buried in permafrost. And then 52,000 years later, it's uncovered. And, sometimes you can get, some tissue samples from these woolly mammoths and do ancient DNA studies. But everyone knows of kind of an ancient DNA that the DNA is like tiny fragments, right? It's not big intact chromosomes, tiny fragments, 50 bases. 100 bases. But a basic question you could ask is the geometry of these things preserved, or is its geometry lost? These are not mineralogical fossils, but they are morphological relics, right, of the chromosomes. That's what we're asking. Are there morphological relics of the original chromosomes? Are the tiny fragments of DNA still in the original geometry, or have they lost the geometry? Is the picture on the right, where we're going to be able to see, very nice, very nice IC maps, or is it the picture on the left where the geometry is totally gone? And we didn't know what the answer was going to be. We started off our process in the stupidest possible way, which is, I think, probably how we started off all of our processes. We started to order mammoth materials from eBay, and you can get them. You can get anything on eBay, but it wasn't the good stuff. To be honest, it just wasn't the good stuff, and everything looked terrible. It looked like the pictures on the left. So we said, no, we gotta get real. We got to start working with great collaborators who can get us the best quality samples. Let's try to find the best woolly mammoth on Earth, the best really preserved woolly mammoth on Earth. Let's try to find it, or something close to it. And so to answer this question of whether it's possible that the chromosome morphology is preserved in ancient samples, we teamed up with a terrific team, Marty Renholm, a wonderful physicist and computational biologist, and Tom Gilbert, really a pioneer in the ancient DNA space. We started working on this. It's like 10 years it took us to do. I mean, it's just a very long time. But it was really a great collaboration. This was work all led by Marcela Sandoval Velasco, Olga Duchenko, Juan Antonio Rodriguez, Cynthia Perez Estrada. Really, really terrific. You can see an early version of this on bioRxiv. We developed this paleo-Hi-C protocol, and we said we need to apply it to the very best samples. So we went on a mammoth hunt, and here is Valery Plotnikov, the Sacha Academy of Sciences, and Dan Fisher, our collaborators at UMichigan, and they found a really, really, really good mammoth sample in Belogoraya in Yakutsk. How good is it? Well, you can see it's still got the hair. Right, it's a big chunk of a woolly mammoth skin. It's still got the hair intact, so it's in pretty good shape. And actually, with a number of our collaborators who did some histology on this, right? So looking at, kind of zoom in, you see the big chunks of it. You can see sort of the hair, and if you look histologically under the microscope, like the skin was still kind of intact. The dermis. You could even see, this is the very first time it's actually been seen. You can see hair follicles. Is a woolly mammoth hair follicle. It's the first micrograph ever taken. And then if you look at the muscle tissue, you can see the myofibers and the long lines of nuclei along the myofibers. So we were like, holy smokes, this thing, it's preserved from the sort of macro multimeter scale down to the multi-micron scale. Now past microns, it's not well known how much ancient morphology is preserved in any form, right? There's some ideas about different kinds of fossils that might exist, but it's kind of hard, and people don't always agree. A lot of it is very, very, very unclear past the micron scale. So we said, well, maybe the morphology of the chromosomes is present at scales past the micron scale. Well, we were able to do a Hi-C experiment successfully on this sample. Here you can see it. This is the Hi-C experiment for the woolly mammoth. It enabled us to actually assemble the mammoth genome. I'm not going to get into the exact details how we did the assembly. We had to use some tricks that we don't normally use because there was so little data. But the spoiler alert is its chromosomes look a lot like those of an elephant. Also, a disclosure, I'm on the scientific advisory board of Colossal Biosciences, which is attempting to bring back a woolly mammoth, and they reached out to us actually as they were in early stealth mode. They knew about this, so we shared all of our resources with them, and I'm on their SAB, and that's a very interesting project. But anyway, we were able to assemble the genome of the mammoth. This is the first time that a genome had been assembled for an extinct species, indeed the first time that any sort of karyotype had been generated for an extinct species. And it highlighted the preservation of these chromosome territories, structures that are only a micron or two in size, right? You can still clearly see them. We can use physics to take these maps and sort of reconstruct how the mammoth chromosomes were folded. And here's like 3D, sort of reconstructions of the detailed folding of the chromosomes. So we felt pretty good about that. We asked the following question. We said, can we see which genes are active? Now remember, in the human genome, if I zoom in on a chromosome, what I see is these patterns of these kind of plaid-like, checkerboard-like patterns of activation and inactivation, right? Because the segregation of open and closed chromatin. Now, in the study of, sort of human Hi-C, this is sometimes and people know this, but sometimes it's not the main reason people do it, because there's so many other ways to check gene activation in a modern sample. Hi-C is not your go-to way to do it. But for the woolly mammoth, there is no genome-wide epigenetic data of any sort available at the present time. And so it would be really great if we still could see this checkerboard pattern of activation and inactivation. So let's zoom in on one of these chromosomes. And lo and behold, it's still there. Now, I do want to emphasize that the number of contacts that we extracted in this map is much lower than what you would typically get in a modern Hi-C map. So it's a couple million. This is a worse map than the maps, for instance, that we published in 2009, the very first Hi-C maps. It is a worse map. And so you're going to notice that sparsity when we look at these things. Nevertheless, the pattern of compartments is unmistakable here. So that made us feel like sort of pretty interested. Hey, check it out. We can see the compartments. Those are usually associated with activation, inactivation. So we immediately asked ourselves, like, what can we do with this? Let's take this out for a spin. Now, one issue is, the resolution is low, and it turns out one of the ways that you can deal with this is less often used these days because the maps are deeper, is you can calculate autocorrelation matrices of the original contact matrix. And this really pumps up the signal. So here, red, indicates that two rows have highly correlated contact profile genome-wide. Blue indicates that they're anti-correlated. And you can see it sort of recapitulates the pattern, but actually at finer resolution. Enables finer resolution, as we'll see in a moment. So that was cool, right? We saw something that looked like compartments. We said, well, are we sure it's the compartments? So, well, how would you look at that? One thing is you can calculate sort of the first principal component of the eigenvector of this autocorrelation matrix, and it typically points, sort of one way if you're an active chromatin, another way if you're an inactive chromatin. So this is sort of a genome-wide epigenetic map of some sort, right? And this is a sort of standard methodology for modern Hi-C. So that's good, but how do we know that we were actually getting something truly epigenetic? That's what we were really wondering about. Well, the first thing we said is, like, are we getting anything sane at all? So we took also a skin sample instead of from a mammoth. We took a skin sample now from an Asian elephant, its closest living relative, and you can see that the autocorrelation matrices, they look really similar, and the contact matrices look really similar. So that actually made us feel pretty confident that we are seeing real compartmentalization, not some other sort of artifact. And when you looked at these eigenvectors, they're really, really similar. So we said, hey, check it out. We really seem to be picking up this compartmentalization. But the key question, right, the key question that would be so enabling is the following, right? Is it the case that what we're seeing, we still have the capacity to see cell type specific epigenetic features? Because that means that we can start to do epigenetics and, sort of even transcriptional, we could use these eigenvectors, these principal components, perhaps as transcriptional proxies, as proxies for RNA-seq and other kinds of things that you can't do in woolly mammoths. Well, check this out. So we did an experiment. We looked at Asian elephant skin, and we said, does the Asian elephant skin look more like Asian elephant blood or like woolly mammoth skin? And lo and behold, the overall pattern looked a lot more like woolly mammoth skin than like Asian elephant blood, despite the fact that this is like some ancient 52,000-year-old sample. And you can see in the autocorrelation matrix, right? Look at this bright red block over here, right? Is all active chromatin. And, look in the mammoth, it's perfectly well preserved. Even in blood, it's not preserved because, the parts of the genome are activating and inactivating. So it seemed to be cell type specific. So we said, okay, it's good. So we got a bunch more tissues. It's hard to do, right? Because, elephants are endangered, but we had our DNA zoo network, so we were able to get more tissues. Now we did a larger panel. Here we're comparing that Asian elephant skin to liver from the very same individual Asian elephant, to blood, to ovaries, again, from the very same individual Asian elephant, to brain, again, the very same individual Asian elephant, and also that mammoth skin. And lo and behold, the eigenvector, this principal component that corresponds to activation and inactivation, was closer for Asian elephant skin to woolly mammoth skin than to any of these other tissues, despite the fact that, some of these tissues are being drawn from the very same modern individual. So it highlights that we're capturing cell type specific, the residues of cell type specific transcriptional activity. And this, in fact, can actually help you nominate differentially active regions. So here I'm just showing the whole eigenvector, sort of for the whole genome. Asian elephant, woolly mammoth, they look very, very similar, but they don't look similar absolutely everywhere. So here's like BARX2, this is a gene that has actually a very strong hair phenotype in mice. And you can see right at the gene BARX2, there's this inactivation in the woolly mammoth that isn't seen in the Asian elephant. Here we're looking at DUSP10. This is associated with development of brown fat, and again, differentially regulated in Asian elephant versus woolly mammoth, which makes some sense. Woolly mammoths are in the cold. So, you expect things like, cold adaptations, hair adaptations. I can tell you a lot more about this, but I'm running out of time. So I want to kind of loop back to the very beginning and ask, are loops preserved after 52,000 years? And this is now going to really test us, because loops are held together by cohesin. Those cohesin rings are about 50 nanometers in size. So remember, I showed you originally we're having this preservation at the multimeter scale. In the histology, we saw the multi-micron scale. Now we're starting to ask for a feature where, if you jiggle things around even a few nanometers, right, you're potentially going to break the loops. So there's a real test. Okay, so we looked at the mammoth map. Is there anything that looks like a loop? Obviously not. This map is horrible. Sorry, I need to remind you that it's a horrible map by the standards of modern maps. It's terrible. So that's not going to work. It's far too sparse, among other things, to see individual loops. Now, so what are we going to do? Well, it turns out you can study loops in aggregate in very sparse Hi-C maps using the following trick. This is something we developed in our 2014 paper, Introducing in situ Hi-C. So here's how it works, right? On the left, I have a good map. On the right, I have a bad map. I can't see loops because I'm too zoomed out. But now let's zoom in on a region where I can see loops. Look, on the left, I can see the bright loops. And on the right, I cannot see the bright loops. So what can I do? Well, here's what I can do. I can cut out little submatrices of the contact matrix around each loop. And I can take all these submatrices on the left and I'll sum them. And then I'll do the same thing on the right. We call this aggregate peak analysis. Look, you see the aggregate signal of all the loops on the left. Look at that. Lo and behold, this is the very first, on the right, this is the very first Hi-C map we ever published. And you can see that enrichment right in the center. And in fact, so we call this aggregate peak analysis. In fact, for the loops that we annotated in the first paper about loops, those loops were reflected in every human Hi-C data set that had ever been published to that time. So here's all the APA, all the aggregate peak analyses. You can actually gradually see how the technology is getting better and better in the hands of different labs as this, these features get more and more robust. But it's always there. So I said, what about aggregate peak analysis? Maybe we can do that here. So we called loops on an African elephant in fibroblasts. And then we looked in the woolly mammoth, and lo and behold, that aggregate signal is there. And if you compare that to the aggregate signal in the Asian elephant, it's the same level of intensity. So what's interesting is even the relative intensity of the loops in this mammoth data is just as strong as it is in Asian elephant data. So what that's telling us is, look, and I want to be clear, and I should have mentioned this right at the outset. When you do a paleo Hi-C experiment, 99% of what you're going to get is contaminants, right? There's only those contacts that are super useful. You're not going to get a ton, right? Here we have a few million contacts in this paleo Hi-C map of the woolly mammoth. But the contacts that we're getting, they show the exact same enrichment for the loops as you get in a modern map. In fact, they show many of the statistical properties. They're almost indistinguishable from modern maps in terms of their statistical properties. So what that's telling us seems to suggest that the loops are still down there, and we suspect the proteins are still down there. I mean, in fact, we think the loops are down there, and how could that be? We suspect where the proteins have gone. We suspect the CTCF is still down there, and the cohesin is still down there. Here we did actually a more extreme version of this. So we said, let's take the human skin loops that we annotated as part of the ENCODE project, and a large project I didn't have any time to tell you about. We annotated loops in like every tissue in the human body. So we took loops in human skin from the ENCODE atlas. We looked for the aggregate signal in Asian elephant skin, right? Beautiful. They're conserved from human to elephant. Very nice. And then we looked for that signal again in the mammoth skin. And it's preserved, and the overall enrichment is identical. Here you can see 2.05 fold for the Asian elephant in the center versus in the lower left. That's how we compare. Here it's 2.08 fold. So really well preserved. So I want to actually kind of summarize, right? In total, what we're observing here is the preservation in these samples of morphological features across 8 orders of magnitude, right? from a 3 meter full carcass in the case of Yucca, another mammoth I didn't have time to tell you about, down to histological features at the scale of microns, and down finally to loops at the scale of tens of nanometers, all preserved. So what we're seeing really is these morphological relics. Some people are sticklers, so fossil means mineralized, okay. But I say morphological relics or indeed morphological fossils of ancient chromosomes down to nanometer scales. So let me tell you just a few conclusions. The shape of a DNA in a cell controls the cell's function. We can read the shape of DNA using high-C contact maps. These allow us to see loops and looping mechanisms like extrusion that turn genes on and off. By measuring the shape of DNA, we can accelerate what the Human Genome Project did and get a genome in a few days for a few $100s for any species that we like. Long after we die, the detailed patterns of our gene regulation may survive still in the shape of our DNA and these fossils of the ancient chromosomes. And this may be useful in bringing back extinct life forms like the woolly mammoth. And finally, 70 years after the discovery of the double helix, we are just at the dawn of physical genomics. We're just starting to understand the shape of DNA. I want to thank a huge number of people who contributed to this and our many funders from our group looking at all these problems. A lot of the work I started in the beginning was led by Suhas Rao, Miriam Huntley, and Adrian Sanborn on development of in situ IC and the proposal that loops formed by extrusion. The work on DNA across the tree of life was all led by Olga Dubchenko in my group. She's an incredibly talented, now assistant professor working in my lab. And she's absolutely wonderful and brilliant and really has made, just contributions that have really, I think, transformed the state of genome assembly from something that took a very large team to something that's trivially easy. And we work with many DNA zoo sites all over the world that contribute samples, for all the many genomes that we assemble and release, contributed many of the elephant samples that we use. And finally, work on the shape of ancient DNA and the fossils and chromosomes was a wonderful collaboration over many years with Tom Gilbert, Marty Renum, and Marcella and Tom's lab, Juan Antonio Rodriguez in Mark's lab, Marcella and Tom's lab, and Cynthia and Olga in my group. Yeah, it's just been a really, really exciting time. Finally, the ultimate manuscript, you can see an early version of it on bioRxiv, has many authors and contributors. And yeah, so the story of the shape of genome is in its early days, and there are many things left to discover. It's been a real pleasure to chat with you about it.